Okay, I think none of you have question about the library preparation, so we continue the next part. Okay, now the next part is NGS platforms. After library preparation, we dive into the NGS platforms. Okay, because after library preparation, we amplify we amplify the DNA fragments. Okay, now we sequence the DNA fragments. There are multiple types of NGS platforms, in which first of all is Illumina, that uses the ion conductance approach to sequence the data, because during nucleotide when nucleotides are attached with each other, the H hydrogen ion is released and pH is low. Okay, pH is reduced. We measure the pH. Okay, during nucleotide addition we measure we don't measure, actually the Illumina is measure the pH. By measure the pH, they infer there is actually A, T, G or C. Okay, by this they know about is there is either A, T, G or C. It's actually the machine in which we sequence the data or sequence the DNA. It's a Illumina. And the ion torrent uses the semiconductor technology. Sorry. In Illumina, we use the reverse dye. Okay. In the reverse dye terminator, when nucleotide is added, our terminator is added. Okay. The terminator is added and image is taken. And after taken the image, the terminator is removed and the next nucleotide is added, and again the terminator is added and image is taken. The steps is in that way. By Illumina we use reverse dye terminator. In the ion torrent we use the technology of ion conductance. Okay, in ion conductance the hydrogen ion is released, and that hydrogen ion when released, pH is dropped and that pH is measured. That is actually the machine of ion torrent. In PacBio, the SMRT sequencing is occurred. Okay, no termination is required, and the real time sequencing can be measured by PacBio. No termination. This is actually the machine. But in the Oxford nanopore, no DNA polymerase is needed. Okay, no DNA polymerase is needed means the actual sequence is infer. Okay, the actual sequence is infer and don't need DNA polymerase like this type of machines is used in Oxford nanopore. So we talk about the ion conductance sequencing that are used by the Illumina. Okay, I think so. Okay, we are here. Okay, in ion conductance, as we all know, for ion conductance sequencing, indexed libraries are amplified using primers. Okay, that is immobilized on the microparticle beads in an aqueous oil emulsion using adapters on the library fragments complementary to the immobilized primers. Okay, now we have DNA fragments. Okay, adapters are attached with it. Actually, the beads or the gene chips are used in which the immobilized primers are attached. Okay, like these are the immobilized primers. The DNA fragments are attached with it. Okay, the DNA fragments are attached with it. And by this, the bead carrying the amplicons are placed on solid surface gene chip. Okay, that is the gene chip. Primers are attached with it. Okay, and the DNA fragments are ligated to the primers, and that DNA fragments then amplified, okay, by PCR. And when they are amplified, and after amplification, sequencing occurs. And when nucleotides are attached to each other, the H hydrogen ion is released and pH is reduced. When pH is reduced, we measure the pH. Okay, this is actually the principle of ion conductance sequencing. The captured fragments are subjected to the addition of the nucleotides in a predetermined order. Okay, the captured, that are the captured fragments to the primers, are subjected to addition of nucleotides. Okay, the nucleotides are added with it. Nucleotides are added with it. If the nucleotide is complementary to the sequencing template. Okay, if the nucleotide, the first nucleotide, is complementary to the DNA sequence, the DNA polymerase will catalyze the formation of the phosphodiester bonds. Okay, the phosphodiester bond will add. By this, H positive is released. As here, a hydrogen ion is released upon the formation of the phosphodiester bond. The hydrogen ion will lower the pH. Okay, that hydrogen ion will lower the pH of the reaction by specific amount of recorded by the sequencer. Okay, by H positive, the pH is dropped, and that is measured by the sequencer. The reaction occurs in hundreds of the thousands of the time. Okay, that reaction occurs in the hundreds and thousands of the time, producing a sequence information from the billions of the library fragments. Okay, and by this, sequencing is done. In the reversible determinator, that is actually by alumina, in which the captured or amplified fragments are hybridized to the immobilized primers on the solid surface. Again, this is a solid surface, that are the primers. Okay, and DNA fragments are joined with it. The fragments are amplified by branch PCR into collection of products or colonies. Okay, the branch PCR is used and it creates a colonies. Okay, that are actually called polonies. The polonies are sequenced in place by sequential addition of fluorescence labeled nucleotides. For example, it is a DNA and its a nucleotide is added, and a reversible dye terminator is added. The fluorescent dye is added with it. Okay, the image is captured here. Okay, the image is captured here, and that reversible dye terminator is released, and the next nucleotide is added. If nucleotide is complementary to the template, okay, if the nucleotide is complementary to the sequence, DNA polymerase will extend the primer. Okay, the primer will extend it. As in the Sanger sequencing, each nucleotide is labeled with specific color or floor. Okay, nucleotide is labeled. An image is taken, as we all know, image is taken of the flow cell after each nucleotide addition, recording the presence of each added nucleotide, color and location. Okay, the color and the location of the nucleotide is recorded. After image, the fluorescence are removed. Okay, the dye is removed and the next nucleotide is added. Simultaneously, hundreds of thousands of polonies are sequenced in this way. Okay, anyone has question about the NGS platforms can ask me in the chat box. Do you have any question about NGS platforms? Can ask me in the chat box. Okay, after 20 minutes this call will be ended. Okay, and I will share the link to all of you to join at 10:50. Okay, at 10:50 join the new link. I will share in the chat box. Join this link at 10:50, means after 20 minutes. Not yet, after 20 minutes join that link. Okay, I think some no one has question, so start continue the next part of the NGS. Okay, the next part of the NGS is sequence quality and annotation. Okay, actually the sequence is done. Now we have to check the quality of the imaging or the conductance. Okay, the quality of the imaging in the actually reverse determinator or the conductance, like in the ion conductance, we check by giving the Fred score. Okay, like the Fred score is 1, it means there is a error 1 or of 10. Okay, in the 10 nucleotides there may be a 1 error. If the Phred score is 10, there is a 1 error in between the 10,000 or maybe a 100,000 base pairs. Maybe there is only a 1 error. If the Phred score increases, the error is decreased. Okay, a sequence is then compared to the reference sequence. Okay, the sequence is compared with the reference sequence. The next step is variant identification based on the comparison with the reference sequence. Okay, at the end we check the is there is any uh, there is any mutation or not. Okay, as there is no alignment there, it means there is a mutation. That is also checked by bioinformatics tools that we talk about is in the data analysis. Okay, SNVs are identified in some program based on specific range of expected allele frequencies. Okay, variant allele and the reference allele. When we compare it, the test sample or the variant sample sequence with the reference sequence, we check there is any SNV or SNP. The SNP is the single nucleotide polymorphism that is present greater than 1% in the population, and the SNV is also the single nucleotide variation that is present in less than 1% in the population, but it doesn't cause the disease. Okay, the individually it doesn't cause the disease. Indels means the insertions and deletions can be identified by the realignment. Okay, when we align it, we can also check there is any deletion or not. Okay, if there is a deletion, it can also check by realignment. Okay, now we annotate it. Okay, each variant is not biological clinical consequences. Okay, I think there is a okay. As we all know, the all variants is not cause the disease. Okay, all variants doesn't cause the disease. Okay, some variants are synonymous or silent, that doesn't change the amino acid, so doesn't cause the disease. So protein sequence is not changed. Okay, some may be caused the SNPs that are not involved in causing the disease. Okay, so annotation is very important. Okay, why important? Because we use it to identify the critical variants. Okay, that variants that cause the disease. Okay, and we filter out the other variants that doesn't cause the disease. Okay, that doesn't cause the disease. That all done by bioinformatics tools that we'll talk about in the next part. Okay, there are several components of annotation. Maybe a location of the variant. Okay, the location is very hotspot. Okay, if there is a variation in that hotspot location, it may cause disease. Okay, the variant change is actually a transition, or it may be a transversion. Yes, Abdul Wahab. Yes, Miss Abdul Wahab. Is there a question?
No, I'm okay. No question.
Okay, okay. Okay. Now we check the genetic state. Okay. We check the mutation. Is there mutation cause the disease or not? The quality of the variant code. Okay. We check the Fred score. If the Fred score is very low, there may be error. Okay. If there is an error, the probability of causing the disease is decreased.
Sorry. Ali, can you explain Fred score again?
Okay, okay. Actually,
Mm.
the Fred score is given to the every nucleotide present in the sequence. Okay, every nucleotide of the sequence having a Fred score. In by Fred score, if the Fred score is 1, okay, uh, if you if you your question is about how Fred score is given to the nucleotide, actually we compare the test sample or the test sequence with the reference sequence. Okay, by this we give the Fred scores, and by using the coverage. Okay, we, what is coverage? Means how many times the variant is again and again same. Okay, if one time A is produced by the one DNA fragment, in the next step again A is there, again A is there, again A is there. It means the coverage of the variant is very high. Okay, by this we give the Fred score. If the Fred score is high, if the Fred score is high, the in the error in the sequence is low. Okay, if the Fred score is high, the error in the sequence is very low. Hope so you understand it. Okay,
Yes, yes.
thank you.
Thank you.
Now, welcome. The variant type. Okay, the variant change and the variant type. We could talk about the transition. Okay, means the purine is converted into purine, and in the transversion the purine is converted into pyrimidine, and the pyrimidine is converted into purine. In the genomic position, it's also about the location of the variant. In the genomic position, the position may be is a hotspot. Okay, the position may be a hotspot that actually cause the disease. We check it, and in the annotation the confidence in the variant call is determined by quality and coverage. Okay, by checking the quality means the Fred score, and by coverage is the coverage is the number of times the region containing the variant is sequenced. Okay, from the independent fragments. By again and again fragments, the variant is again and again sequenced. It means the coverage is very high. The chromosomal and sequence location of the variant in the context with the reference sequence is identified. Okay, the location of the variant and the genomic position can also be identified by the reference sequence along with the type of the variant. Okay, for example, it's a test sequence and this is a reference sequence. The location of the variant, it's maybe there, it's maybe there, it's maybe there. It's also identified by compare it with reference sequence. Okay, now we filter it. The variant is subjecting to filter. Maybe there is a okay? There is maybe SNP that doesn't cause the disease. Okay, the variant may be a genetic or a somatic. If it is a genetic, it can be inherited. If it is in a somatic, it doesn't inherit it. Okay, it's also checked by annotation, and we filter out it. For variants in introns, any effects of the splicing or recessed may be the change in the introns. Okay, that doesn't cause the disease or that doesn't cause any clinical variations. Also filter out. Okay, that all are done by using the polyfen and shift bioinformatics tools. Okay, by using the bioinformatics and shift tools. Okay, we check. Okay, after 7 minutes this meeting will be ended and the new please join after 2 minutes. Okay, after 2 minutes we join the new meeting, so please remember it. Okay, the algorithms means the sets of instructions like polyfen and shift is used scores to indicate the degree of damage to the protein structure. Okay, like if the variant cause the amino acid change. Okay, by the variant the amino acid is changed. If the amino acid change, protein is changed. So we check how much the protein is changed or how much the protein is damaged. If the damage is high, the score is high. Okay, the score or the Fred score is high. The Fred score is given by using the bioinformatics tools. Variants that remain after the filtering may be annotated by searching in the disease specified databases. Okay, after filtering out, there may be variants that are not filter out. Okay, that are not filter out, also checked by using the different types of databases. That variant may cause cancer. Okay, that may cause somatic mutations. Okay, that may cause the infectious diseases. Okay, that also checked by using the bioinformatics tools. Okay, now do you have any question about annotation and filtering? Can also ask me. And now we shift to the next Google Meet point because the time is almost out. Okay, now please left the meeting and join the new link. Please left the meeting and join the new link. Heba Muhammad and Saad, please leave this meeting and join the new link. Mubashir, join the new link. Okay, welcome back to the NGS Workshop. Hope so you all hear me. If anyone has question about NGS, annotation and filtering, can ask me in the chat box. And after this, we continue. The last part of the NGS is data analysis. It's actually about the bioinformatics tools, and actual the data analysis will be done. We'll talk about the data analysis of the NGS in the next workshop. But we'll talk about the overview of the data analysis of the NGS in that part in today's workshop. Okay, I think so. No one has question about the filtering and annotation. So we start or continue the next part of the workshop. I am gonna share the screen to all of you. Hope so you all see the screen. Just wait. Now you see the screen. Okay, now we talk about the last part of the NGS is data analysis. Okay, after we get the sequence or the data, okay, now we interpret it. Okay, like we check the sequence quality, we check the annotation, we check the Fred score by using the bioinformatics tools. Okay, there are multiple types of the tools for NGS data analysis. Okay, it's main the main and most important tool of bioinformatics tool is Galaxy. Okay, the Galaxy database, in which we check the Fred score of our sequence. Okay, and annotate the variants. Okay, the variants calls can be annotated by using the Galaxy. We also use the BWA, we use the SATAR, the BED tools, the GATK, CLC, base space, SAM tools, plotting. Every tool has a unique purpose. Every tool has unique purpose and functions. Okay, maybe some functions are aligned with it, maybe some functions are overlapped. Okay, but every tool has a unique purpose as a characteristics. Okay, first of all, when we take the sequence, okay, for example it is alumina. Okay, the sequence or the data in in the sequencer is available in .bcl format. Okay, .bcl format. we have to convert we have to convert fast queue, okay, into fast queue sequence, into fast queue file. Why into fast queue file? Because by the fast queue file we get the Fred score. Okay, by fast queue file we can get the Fred score, as it's a fast queue format in which there is a q score. Okay, there is a q score, that is the quality score or the Fred score. There is a base, maybe a t. Okay, the reference is q, that is a. Okay, maybe so there is a variant. Okay, and the Fred score is there is 25. There is a label for the sequence, and this is actually the sequence, and this is the Fred score. That was called there is a Fred score. Okay, like there is a Fred score is 10. Okay, for example, the Fred score is 10. There is, it's actually a formula. Q is the Fred score. Okay, and the P is the error. If the Fred score or the Q is high, the P is low. Okay, if the Fred quality score is 10, the probability of incorrect base call, means the incorrect base call or the error, is 1 in 10. Okay, so 1 in 10 means the base call accuracy is 90%. Okay, it means there is a 10% probability, there is a 10% probability of error. Okay, there is a 10% probability of error. If the Fred score is 20, so the probability of incorrect base call is 1 in 100 and the accuracy is 99%. If the Fred score is 60, the 1 in 1 million, the 1 in 1 million probability of incorrect base call. Okay, the accuracy is 99.999% accuracy. Okay, so the Fred score must be high. Okay, the Fred score must be high. So by this, the incorrect base call, the probability of incorrect base call is very low, and by this the accuracy of the NGS or the sequence is very high. And it's a principled a beautiful rainbow. Actually, it's a DNA fragment. Okay, it's a DNA. Okay, and we make the DNA fragments. Okay, that DNA fragments is then aligned. Okay, that DNA fragments are aligned at the end. Okay, that is show like this, and is uh, gives us the base quality. Okay, this gives the base quality as a fast QC report. Okay, because by the fast Q file we get the Fred score. If the Fred, like this, the Fred score is 0 0 to 40. Okay, 0 to 40, and the position on the read is the base pairs, is a 1 2 3 4 5 6, like this. The base call or the Fred score of these twos are very low. Okay, and the base call of this is very high. The base call of these are very low. The base call of this is somewhat good, but the base call of the Fred score of this is very good. So can also be checked by using the fast QC report. Okay, it's also a tool of NGS. And by using the UCSC genome browser, we can align or compare the reference sequence with the test sequence. Okay, it's a test sequence, it's a reference sequence. Okay, we compare it. As we check it, the all sequence is aligned well. Okay, the whole sequence is aligned well, but there is a mutation. Okay, there is a mutation. Wherever the mutation, there is a different color. Okay, there is a different color, there is a C. Okay, the actual reference sequence has T. Okay, the actual sequence has T, but the mutation the test sequence or the variant sequence has C. Okay, as we see, there is a C, and the Fred score or the quality or the coverage is very high. There is in this fragment there is a C, there is also a C, there is also C, there is also C. It means the coverage is very high and the Fred score is very high. It means the T is changed with the C, so there is a mutation. Okay, there is a mutation, and that mutation may be cause a somewhat disease. Okay, that's end of that workshop. Thank you for being part of this wonderful journey. Follow for further workshop announcements. Please connect with me, follow me on LinkedIn, and subscribe on the YouTube channel, that is Aritur. And as you all know, there is my email and WhatsApp channel, and you can also have any query, can also contact with me on by WhatsApp on this number. Thank you so much.
Thank you very much, Ali.
It's my prayer to all of you. Thank you.
Thank you. Thank you.
Thank you, sir.
Thank you for joining. I will also add all of you into the WhatsApp group. In that WhatsApp group, I will share the next workshop time. Thank you so much.